Okay, so I just want to do an attendance check real quickly here. At this time, please type present for your attendance. If you did not sign in with your full name, be sure to include it in the message. So it is officially 1:15 now, and we'll go ahead and start in earnest here. I'll wait for a few more of you to check in. I'll try to check in a little bit later for those who arrive a little bit later. For those of you who just arrived, we are checking in now at this time for attendance. Please type present for your attendance. And again, if you did not sign in with your full name, please make sure your name is associated with that. Okay, so let's go ahead and get started here. So what we are going to do today is we're going to talk about the biology of behavior. But before we start there, let's go ahead and cover some of the information that we covered in the last session. Before I really get rolling here, are there any questions at this time before I get started? You can chat with me or just speak up. If not, we'll go ahead and start with the session. Okay, let's go ahead and get started then. So in the last session, we talked about statistics, and this is the analysis of numbers that are generated for a variety of reasons, for surveys, for the research that we've conducted. We see that statistics are involved in research, and we'll see that throughout this course, but also we wanted to emphasize during that last session as well, is that statistics are also important for real life, your everyday life. You run in statistics all the time as people present you with information to try for you to purchase something, a product or service, or to believe in a certain type of belief that they want you to have in your mind as part of your opinion. We also saw too that we can mislead people with statistics. We can do this either intentionally or unintentionally, but the idea is that this ability to mislead with statistics is there, and we see it all the time. And so that's why you need to be critical consumers of information, and hopefully this class will help sharpen those type of skills so that you can recognize some faulty ideas that are being presented to you wrapped up in statistics. So that's one of the things too we want to cover in that last session as well. Then we looked at how it is that we can describe the data. How is it that we can look at the data and get some understanding of what the data is, what it looks like, what it feels like, so to speak. And one of the metrics that we can use is that of central tendency. With central tendency, we talked about the mean, the mode, and the median. The mean you're very familiar with. That's the average, where you add up all the numbers that have been generated by subject or whatever data you have, and then you divide it by how many there were. Okay, the sum of the numbers and then how many there were. And that gives you the essence of what the data looks like, at least the central tendency of the data. We also noted that sometimes, because of what we call outliers, extreme scores, either too low, 0, or negative something, or something too high, like millions of something, where that's not what is normal, then that can elevate the average too much and give us a misleading information in regards to what the data actually looks like. So we have the mode. A mode is another way to describe central tendency, and that is the most frequently occurring number in your data set. And then we have the median, another way to describe it, where you just rank from low to highest numbers in your data set, and you select that middle number that represents the median. Now, along with central tendency, we also talked about how the data varies. That's also important to understand as well. That's variation. We talked about range, which is a crude estimation of variation, where you look at the difference between the lowest number and the highest number, and that just gives you the range of the data. A more precise way of looking at the variation of the data is using the standard deviation, where in essence it takes like that average and then looks at that average compared to each one of the data points in your data set and comes up with a calculation that gives you the standard deviation. How do these numbers vary in a standard format as opposed to just looking at the range? Then we introduced the idea of the normal curve, sometimes talked about the bell curve or the normal curve. Looks like a little bell up sitting on the platform. And the bell curve basically describes this idea that in many phenomena in the world, it falls on a normal curve. And what that means is that at the extreme ends, we have a few numbers that are very, very low, a few numbers that are very, very high, but most of those numbers fall in the middle. Okay, we see that with such things as intelligence scores. Some people are going to be subpar when it comes to that. Some people are going to be above par when it comes to intelligence, but most people are going to be in the middle. Then we revisited the idea of correlation, how two variables are associated with one another or how they are correlated with one another. And we introduced the idea of scatter plots, where if we have X variable and Y variable, and we look at how you scored on X and Y and plot it, and you do that for all the numbers in your data set, then what you'll have is a scatter plot. And from that scatter plot, you can get a good idea as to the relationship by using your eyeball exam. Good idea as to the relationship of those two variables. And then we talked about the correlation coefficient again. That's the strength of the relationship between the variable. The sign tells us the type of relationship. For example, if we have a perfect positive relationship, that tells us that as one variable goes up, so does the other variable, and they tend to fall on a straight line. The beauty of that is if you have a perfect correlation, a positive one where one goes up and the other one goes in the same direction, or even a negative one where one goes up and the other goes down. But if it's a perfect correlation, if you know one variable, you can always predict with a great deal of accuracy what the other variable is going to be. So we revisit the idea of correlation, and then we introduce the idea of the significance test or significance testing. With all this research that we're doing and the experiments that we hopefully can conduct, we're trying to figure out, does something cause something else? Now, we talked about descriptive research, we talked about correlational research, and then we talked about experimental research. But when it comes to the p-value, when we significant test something, we have these two things that we need to understand. We have the null hypothesis, and the null hypothesis is which the researcher is trying to disprove. That means that my idea doesn't hold any merit, that there is no relationship, or this variable does not cause some change in the dependent variable here. All right, so that's the null hypothesis. But we also have this alternative hypothesis, which states that the researcher is trying to prove this alternative hypothesis. The example we used in the last session was, I believe it was biofertilizer X. Yes, that was it, biofertilizer X. And the notion was, we believe biofertilizer X will grow plants better than any other brand. Okay, the null hypothesis would state that biofertilizer X is no different than any other brand. The alternative hypothesis, the one that we want to find support for, would say that it does. So we generate the numbers, okay, and we measure the plants and see how they're growing based on biofertilizer X and all the other brands. So now we have these numbers. And then what we want to find out, is there a significant difference, that is statistical difference, between biofertilizer X and what it did to the plants and compared to the other brands? And that's where the p-value comes in. So the significance testing for statistics is this: how likely is it that an obtained result, the data that we've just obtained from doing this research, happened by chance? That it's just chance. There's nothing really there. The null hypothesis is actually true, but the data that we got just happened by chance. And that's where the p-value comes in. The p-value is the probability of obtaining a test result at least as extreme as the results we actually saw, we actually observed, under the assumption that the null hypothesis is actually correct. Okay, so when we have a p-value of 0.05 or greater, is basically saying we have 5 chances out of 100 or so that we could have gotten this data by accident, and biofertilizer X doesn't do any more than anyone else. That is what we consider to be insufficient evidence against the null hypothesis. So we would not say that biofertilizer X does indeed cause better growth in plants. If we had something that is between 0.01, 1 out of 100, and 4 times out of 100, less than 0.05, we would have some evidence that the null hypothesis is not relevant in this scenario, and we have some support for the alternative hypothesis. But what we're really looking for is a p-value that's less than 0.01. Strong evidence against the null hypothesis, meaning there is no difference between biofertilizer X and the rest. So we have support that our biofertilizer X that we developed does indeed work. So what it comes down to is the p-value is the end result of a lot of the stuff that we've talked about so far, trying to say, are we on the right track in a sense? So that's what we talked about last session when it comes to statistics. And then we sort of shifted gears a little bit to talk about APA for academic writing. It shifts gears only a little bit because I want to emphasize this. American Psychological Association writing is designed for researchers because we don't want to mislead people. We want to make sure people have all the information and data possible for them to do their research. If somebody's reading a paper that you have written, that means they're interested in the topic that you've just written about, that you've researched. Okay, so that's part of the whole research tradition, that you write down everything that you did in your research study in a certain way so those people who read it will understand what they're looking at. So I wanted to highlight last class that there are resources on Canvas, videos, really short videos for the most part, that talk about plagiarism, about paraphrasing, about citations and things of that nature. I highlighted the importance of references and in-text citations. So when you're writing a document, writing a paper, you've researched and you're writing something down, it is going to be assumed, if you are the author of that paper, that the things that you write down are yours, your ideas, your concepts that you came up with, unless you cite it somehow. Okay, and basically what it comes down to, when you write something and then you have an in-text citation, it's like a big finger saying, hey, people who are reading this paper, I didn't come up with this particular idea, this person did. And it usually has a name and a year associated with it. And what that says is that now that you see this in-text citation with this name and this year, you can go back to the last page of this document, the references, and be able to find out more information about where I got this information from, and they can follow up on it if they want. All right. And then we went into some of the key points of the short paper that you guys will be responsible for, about how are some of the things that you can do about collecting data for your paper, about the importance of the cover page for me particularly, and then the key aspects of identify the source article, the author of it, the organization they belong to, and any other pertinent facts that you may have. Then you summarize that source article, okay? And then you relate the source topics of that article to the psychological concepts that we have covered or will cover that are presented in the textbook, for example. That is a lot of what we covered in the last session. Now, before I get going to the new topic here, are there any questions you have at this time? Okay, so with no questions to have there, let's go ahead and move on. So today, what we're going to be focusing on is the biology of behavior, okay? We have behavior, we have mental processes, and so we're going to be looking at the root cause of this, and that's going to be our biology. I start off this section by asking this: where are we located? Where are we located? And what I mean by that is this: searching for the biology of self, who we are. Is our identity in the heart, in the brain, or in the whole body? Biological psychologists explore the associations between body, mind, and behavior, okay? But it should be noted that everything is connected, okay? The body is connected to the brain, connected to what we call the heart or the spirit, if you will. The idea is that I've known people, and I've done it myself sometimes, that you worry about something so much that you make your body sick, okay? And again, that's something too that we'll brush on a little bit later in this semester. But the idea is that the biology of the self, that's one of the things that we've been asking for a long time. Where are we, okay? And most oftentimes we think about the brain. So that's going to be one of the key topics that we'll discuss today as well. So what we have in mind here, the current class meeting, where we are today, we're going to be discussing the building blocks of the mind, neurons and how they communicate via neurotransmitters. We'll look at the systems that build the mind, functions and parts of the nervous system. Then we'll look at a very important supporting player that we don't hear that much about, and that's going to be the slower communicating endocrine system that utilizes hormones. Then we'll wrap it up by looking at tools for examining the brain and its activities. And then the plan is for next class meeting to talk about the more primitive and advanced brain structures, looking at the older brain and then the newer brain, which we are more familiar with for the most part. Okay, so let's get started. And we start off by looking at the building block of the nervous system. We'll be looking at the structure, the communication, the electrochemical process, and then neurotransmitters. This is it. This is a picture, a graph of the neuron. All right. This neuron has a couple of key features about it, but let me walk you through what's going to happen. First, we have dendrites. Okay, and the dendrites are in the upper part here. Hopefully you can see my cursor. And what they do is they're going to be receiving messages from other cells. So the messages coming from other cells are then going to travel down and make some change in the cell body here. And the cell body is going to be responsible for making sure that this neuron can survive. It's the life support center. So when the dendrite receives a message, then a neural impulse, okay, is going to occur. Think of it as like an electrical charge that travels through the body and through what they refer to as the axon here. So the axon passes messages away from the cell body to other neurons, muscles, or glands. So that's the neural impulse, also referred to as an action potential, an electrical signal traveling down the axon. Now you can see, and this is not the case for every axon, there's this little white little gray type of thing that covers this axon here, and this is called the myelin sheath. It covers the axon of some neurons and helps speed up neural impulses. So it travels faster, okay? So when you have a myelinated or myelin sheath on an axon, it goes really, really fast. So basically, it speeds up. And so certain parts of our body will require the additional speed in order to function ideally. All right. Now, you may be familiar with this or not, but sometimes our body has hiccups. And it attacks our self. It attacks the self. It attacks its own body. And sometimes it will get a message to destroy the myelin sheath, therefore interrupting the neural impulse of certain axons. When this happens, when the myelin sheath is being degenerated by the body itself, that's multiple sclerosis. Okay, so now we have the dendrites receiving the message, the neural impulse happening, traveling down the axon, and then it reaches these things at the very end. These are the terminal branches of the axon, and these form other junctions with other cells and other neurons. By the way, most likely there'll be other neurons over here that sent the message to the dendrite. Okay, we'll see some more details about this as we go. Now, let's talk about the action potential, this neural impulse that travels down an axon like a wave. Okay, so neuron stimulation causes a brief change in electrical charge. If it's strong enough, this opens up these gates to allow positively charged sodium ions to flood in, producing a momentary depolarization called the action potential. So what happens is there's enough stimulation, okay, and then the stimulation is detected, it reaches a threshold, and then all of a sudden we have these positive charged ions flooding into the neuron, okay? The next thing that happens in stage 2, this initial depolarization, the action potential, influences the electrical charge of the next portion of the axon. That's what is meant by it travels like a wave. You remember how fans in a big stadium, when we had them filled up to capacity, they may stand up and put their arms high up, and then they sit down, the person next to them does the same thing. That's what's happening here in a way. So we have the ions rushing in, and then we have ions rushing out, and it's causing a wave to travel down the body of the axon here. All right, so the gates in the neighboring area now open, allowing positively charged sodium ions to flow in and depolarize that area. Meanwhile, other gates open in the first part of the axon, allowing potassium ions to flow out, repolarizing. So as it waves, goes up, and then the one behind it goes down. All right. So at this action potential moves specifically down the axon, sodium potassium pumps in the cell membrane finish, restoring the first section of the axon to its resting potential. So what I want you to imagine here is that there's enough stimulation happening in the body, okay? And what happens is once a threshold has reached, we go up to the graph up top here. Once a threshold has released, there's enough stimulation, and then the neurostimulation occurs, and that's your action potential. Then it goes down after it has done its job and gets to resting potential. Like a wave, it flows through the axon, through the neuron here, until the message keeps going all the way. Then ultimately we see what is going to happen at the other end. So when does the cell send the action potential? When it reaches that threshold. The neuron receives signals from other neurons. Some are telling it to fire and some are telling it not to fire. And that's another key point. All the neurons can do here is say fire or don't fire, fire or don't fire, 1 or 0. Very similar to what we call a computer program, or binary, 1 or 0, fire or don't fire. When the threshold is reached, the action potential starts moving, okay, like a wave. Okay, so it either fires or it doesn't fire. There's no half fire or over fire, no overloading. It fires the same way every single time. Okay, more stimulation doesn't do anything. All right, this is what they refer to as an all or none response. You can't halfway be it. It's just going to be the same way every single time. You can't like be partly pregnant. You either are or you aren't. It's all or none response. So the threshold reached when excitatory fire signals outweigh the inhibitory don't fire signals by a certain amount. Now, somebody asked this at one point. Let me give you a description here. Well, what's the difference between getting punched or getting tapped on the shoulder? Okay, how will we treat that differently? So the more neurons fire when you're punched. So when you get a punch, more neurons are taking place, more firing, compared to when you're simply tapped on the shoulder. So that is going to be sort of how this process works. Now, the action potential travels down the axon from the cell body to those terminal branches that we just saw earlier. All right. And then the signal is transmitted to another cell. All right. However, the message must find a way across a gap between the cells. This gap is called the synapse. Now, an interesting thing, we may have billions and billions of these neurons in our brain and other parts of our body and whatnot, but they never actually touch each other. They never touch. Okay, so it's like, you know, when your kids and your little sister is screaming because their brother is pretending to touch them, okay, but they're really not exactly touching, they're just pretending they're going to touch. Well, these neurons never touch each other at all. Here's a picture of the synapse. So the synapse is a junction between the axon tip of the sending neuron and the dendrite or cell body of the receiving neuron. So the synapse is also referred to as the synaptic junction or the synaptic gap. So let's talk a little bit about what we're seeing in this figure here. So we have this being the sending neuron up here. The action potential is traveling down, and there's these vesicles containing neurotransmitters, and we'll talk more about those in a second. Neurotransmitters. These neurotransmitters are then going to release themselves so they can travel across the synaptic gap here to land on receptor sites of the receiving neuron. It's going to be like a lock and key. So the key is going to be the neurotransmitter. The lock is going to be the receptor sites here on the dendrite here. So once that happens, the message has been transferred. Okay, either fire or don't fire. So the message, and it goes on and does it to the next grouping of neurons that it's attached to or associated with. Now, earlier I referred to an electrochemical process, and let me describe that to you real quickly here. The action potential is part of the electro process. It's a neural impulse, electrical impulse traveling throughout the body. But then it becomes a chemical process, because that's the only way the communication can happen between this sending neuron and this receiving neuron is through the chemical of the neurotransmitter. The pulse, the neural impulse, can't jump over the gap. It can only get to across the gap by releasing the neurotransmitters. And that's what we mean by electrochemical process. So neurotransmitters are chemicals used to send a signal across the synaptic gap. So it releases the neurotransmitter to go here to continue its process. Now, that implies something very important, that there are going to be a variety of different types of neurotransmitters, and we'll see different neurotransmitters will have different effects on the body, and we'll see that in a little bit. Now, once the neurotransmitter has released its, once the axon, the sending axon has released its neurotransmitter, okay, and the process is done, after neurotransmitters stimulate the receptors on the receiving neuron, the chemicals are taken back up into the sending neuron to be used again. This reabsorption process is called reuptake, and this ends the transmission of the signal. So what happens now, that's a complete signal. It's done its job, and now it's reabsorbing excess neurotransmitter back up into itself. All right. Now, I'll take a point here, hopefully it won't be too confusing, to talk about drugs and how drugs can have certain impacts on our behavior. You guys, I'm certain, have heard of drugs like Prozac and Zoloft and Paxil. And tell me through chat if you've heard of any of these. Zoloft, Paxil, Prozac. Okay, good. So what that tells me that you guys are familiar, as most people are, and I excellent, someone says SSRI, exactly. It's selective serotonin reuptake inhibitor. And that's why I just want to point this out real briefly here, is that serotonin is a certain type of neurotransmitter, and we'll talk about those in a little bit. All right. So with serotonin, it makes us, when we have serotonin active in our brain for longer periods of time, we tend to be less depressed. Okay. We tend to be less depressed. And so what they figured out, if we can stop or prevent this reuptake from happening for longer periods of time, and that's what SSRIs do, selective serotonin reuptake inhibitor, they keep the serotonin active longer, then that's the medicine, and that's how it has an impact on our mood. We'll see more about that in a little bit, but I just want to go ahead and point that out to you as we're looking at the synapse here and looking at the reuptake of a neurotransmitter. I want to give you that notion because most of you are familiar with some of those SSRI names like Prozac and Paxil. All right. So seeing all the steps together, we have the electrical impulse or action potentials travel down a neuron's axon until reaching a tiny junction known as the synapse. When an action potential reaches the axon terminal, it stimulates the release of neurotransmitter molecules. These molecules cross the synaptic gap and bind to the receptor sites of the receiving neuron. This allows electrically charged atoms to enter and receiving neuron to either excite or inhibit a new action potential. And 3, the sending neuron normally reabsorbs excess neurotransmitter molecules, a process called, again, reuptake. That is what we're talking about. A very, in a way, elegant and simple process with some complexity thrown in. Now, there are numerous types of neurotransmitters. Okay, we have acetylcholine. This enables muscle action, learning, and memory. Some examples of when they malfunction: when we have problems with acetylcholine neurotransmitters or neurons that utilize that. We have Alzheimer's disease, when the acetylcholine producing neurons begin to deteriorate. We have dopamine, which influences movement, learning, attention, and emotion. And when we have an oversupply of dopamine linked to schizophrenia, and an undersupply linked to tremors and decreased mobility and Parkinson's disease. Then we have our friend serotonin, which affects mood, hunger, sleep, and arousal. In fact, much of the earlier research, is my understanding, serotonin was focused on sleep, and then they discovered the impact it had on mood. So the examples of malfunction of serotonin, an undersupply is linked to depression. That's why we have the SSRIs, to keep serotonin active for longer periods of time in individuals who are suffering from depression. Some drugs that raise serotonin levels are used to treat depression. All right. Then we have norepinephrine, helps control alertness and arousal. Undersupply can depress mood. We have GABA, a major inhibitory neurotransmitter. The undersupply has been linked to seizures, tremors, and insomnia. Glutamate, a major excitatory neurotransmitter involved in memory. Oversupply can overstimulate brain, producing migraines or seizures. Some people who have reactions to MSG that we sometimes see in certain types of food, they have to avoid that. And then we have our friend endorphins. I say our friend because it's very important. Endorphins are a neurotransmitter that influence the perception of pain or pleasure. An oversupply with opiate drugs can suppress the body's natural endorphin supply. The body, and if you've seen any of the videos yet, the brain in particular is nothing but a really big chemical manufacturing facility. It produces all sorts of chemicals. One of them is an endorphin. Endorphins are very similar to drugs called opiates, like morphine and that type of stuff. So when you stub your toe, your body will begin to release endorphins in order for you to deal with the pain. If you do something really good, and I don't do enough of this lately, go out and do some running and exercise quite a bit, and your sweat, your hands are on your knees, you're bent over and you're breathing happy, but you feel good, that's because of endorphins. They've supplied you with a little bit of opiate type of drug, endorphin, in order to make you feel good, which rewards you for doing something the body tends to like. So these are some of the roles of different neurotransmitters. I'll pause there before I move on and see if there are any questions or comments at this time. All right, there doesn't seem to be any chat messages, so let's keep moving on. So what I want to also emphasize is this other idea too. So we have all these different types of neurotransmitters, and we're still discovering what they do and how we can influence them in some way to have some sort of impact on our body and processing. But there are going to be some serotonin pathways. Networks of neurons that communicate with serotonin help regulate mood. So again, we have these types of neurons that are sort of slated for serotonin, and others slated for dopamine. So we have the serotonin pathways and dopamine pathways. Networks of neurons that communicate with dopamine are involved in focusing attention and controlling movement. So again, we are learning quite a bit about our neurons and the path they take within the brain and how they influence our body in many, many ways. So what we've done so far, we've talked about the basic building block of the nervous system, and that is the neuron and how the neuron communicates electrochemically, a little bit about the neurotransmitters. And now we want to talk about the nervous system. And this is broken down into the central nervous system and the peripheral nervous system. Now, you're very familiar with the nervous system. I'm sure you understand some basic ideas about it, particularly the central nervous system. The nervous system is broken down into the peripheral and central. The central nervous system, all that really is the brain and the spinal cord. Now, I say all that really is. It's very important, but it's just made up of the brain and the spinal cord. And that's what we see on the right side of this graph here, this slide here. The brain and the spinal cord. That's the central nervous system. Everything else is peripheral to it, outside of the central nervous system. And we have the peripheral nervous system broken up into the autonomic and the somatic. The autonomic nervous system and the somatic nervous system, we'll see more about that in a little bit, but let me go ahead and tell you, the autonomic nervous system controls self-regulated action of internal organs and glands. Okay, the somatic controls voluntary movements of skeletal muscles. So we have control when I clap my hands. That is the somatic system at play here. Okay, the central nervous system is at play too, because I had the idea and it sent it down the spinal cord to go to my hands to do that. But it controls voluntary movements of the skeletal muscles. That's what its responsibility is. Whereas the autonomic system, this stuff is self-regulated. We don't control it. Some people may be able to get themselves into a certain mindset where they can influence certain types of things, but that's something that is controlled outside of us for the most part. And then we'll talk about the sympathetic and parasympathetic a little bit later as we go. So primarily the way we can look at it is this. The central nervous system, the brain and the spinal cord, is the body's decision maker. So it comes up with the ideas and then sends the instructions down. All right. Now, it should note that the spinal cord is a two-way information highway connecting the central nervous system, okay, and the peripheral nervous system to the brain. So that's one of its important things. The spine is getting information from the peripheral nervous system and sending it to the brain. So the brain can make a decision and send instructions down to the body to do whatever is necessary. Now, the peripheral nervous system gathers information from the body and sends to the central nervous system and decisions out to the body. So the idea here, the peripheral nervous system collects information, sends it up to the decision maker. The decision maker, the brain, makes the decisions and sends instructions down for the body to carry it out. That's basically us and how our nervous system works. Now, there are certain types of neurons. We have the afferent or the sensory neurons, and they carry messages from the body's tissues and sensory receptors inward to the spinal cord and the brain for processing. So when you look at something, okay, you have specialized receptors that will take that information in, okay, and then send it to the proper area inward to the proper area of the brain ultimately for processing. So then you know what you are sensing, for example. Then we have the motor efferent neurons. They carry instructions from the central nervous system out to the body's muscles and glands. So if you see, for example, you're walking around across the street and you see, for example, a big truck burning rubber headed towards you, your brain processes the danger you're in and sends instructions for your body to do something. Okay, so that's what it's doing. It's getting information, getting instructions to do something. And then we have the other interneurons in the brain and spinal cord, and they process information between the sensory input and the motor output. They're going to be millions upon millions of sensory neurons, millions upon millions of motor neurons, and a lot more billions of interneurons. And that's all these things working together for you guys to do your daily activities, to do whatever it is required for you to do. And we'll talk more about the sensory stuff in a later chapter, just to wrap up some of these key ideas. So when we look at the peripheral nervous system a little bit more detailed now, we talked about the autonomic and the somatic. Now let's look at the sympathetic and the parasympathetic. The sympathetic system, its responsibility for us is to get us into an emergency. We're in an emergency situation and it gets our body ready to do something about it. Okay. The parasympathetic purpose is to calm us down, to get us back to green operating status. Whereas the sympathetic arousing, that's when we're in the red zone. Red zone, sympathetic, green zone, parasympathetic. All right. One arousing, one calming. So let's talk about this. So why are we not always aroused or super alert all the time? When you are really worried about something, stressed out about something, you know how much energy that takes? Okay, it can tire our body out. In fact, around the times, there are two times when we normally see students catch colds or flus quite regularly, and that's going to be around midterms and around finals, because you're stressing out a lot. You're maybe not sleeping as well, okay? And your body is not going to be able to defend itself as well when that situation is going on. So the system that we're talking about here is the autonomic system. We have the sympathetic and the parasympathetic. When you sense that you are in danger, let's say you're at an ATM machine, you're getting money out, it's a late Friday night in a somewhat deserted area, but you need some cash real quickly, and all of a sudden you hear a bunch of footsteps behind you, and they start to sound like they're running towards you. Well, what's going to happen is your sympathetic nervous system more than likely will engage itself. It will arouse your body in a certain way. Your pupils will dilate. Your heart will start to accelerate. That Big Mac that you ate an hour or so earlier that you're digesting now, it'll stop digestion. Okay, uh, your liver will stimulate glucose release. Epinephrine and norepinephrine will be released by the adrenal glands. Your bladder may relax a little bit. Okay, so all this is happening in order to prepare you for a very important decision that you have to make. When your body senses danger, it gets you ready to deal with that danger. And that, as you know, is the fight or flight response. Amazingly, the things that it's doing to your body at this time can be used for either one. It can prepare you to fight it out or flight or run away. And once the danger is over, once you're no longer in danger, the parasympathetic system takes over and begins to calm things down, get you back into a normal state. All right. Here, your pupils will contract when they once were dilated, they become contracted. Your heartbeat will slow down. You'll begin to digest that Big Mac you ate earlier. So that's what we see there. And so the body has been trained and conditioned and programmed to act in certain ways like that. All right. Any questions before we move on to the next subtopic here of neural networks? Okay, seems like we're all good here. All right, so that talks about the automatic nervous system. Now, I want to share with you another basic idea too. We have these things are neural networks, okay? And these complex webs of interconnected neurons form with experience. And obviously, when we are younger, oftentimes, it's been going on for generations now, parents try to get their kids involved in a whole host of things. It's not unusual for you to have had, as a parent, the experience of doing some of this or receiving some of this when you were a kid, where you have piano lessons, maybe you have karate or dance lessons, you have some other extracurricular activity, you have hiking club, whatever it is. And what you're trying to do is to get you interested in all sorts of things. But from another point of view, what it does, it starts to develop neural networks. Networks of neurons that are activated when you are practicing your karate or when you're playing your piano. Okay, so all these neurons that are at work when you're practicing these types of things, they fire together and wire together. They become sort of connected in a way. They become a neural network. All right. And these neural networks are activated when needed for action. So you can sit down and then that network for you to play the piano activates and you start to play the piano. Now, again, that's wonderful when you're young, but after a while, if you don't like going to piano lessons, you may stop for a long, long time. And there usually is going to be a time, and we'll talk about this a little bit later as well, that those things that are not being utilized much, they may sort of be pruned away. It's the idea, use it or lose it. Okay, so the idea here is that these neural networks do establish themselves for that particular reason, that they fire together and they sort of become wired together for certain types of activities that you've been involved in, certain inputs, and then certain outputs that it's trying to achieve. All right. Now, another thing that, about the neurons or the interneurons that we mentioned earlier is that sometimes things can happen without our brain having to be involved. Okay. That there are certain types of things that we can act on without the brain's involvement. And we sort of learn about it after the fact. So if you've ever been walking in a kitchen that you're sort of not paying attention and you put your hand down somewhere and you realize that you put it down on a live eye on a stove or something, and you don't realize it yet, but you realize that your hand is moving back of its own accord seemingly, and then you realize what just happened. Okay, because there's sort of a simple natural reflex that occurs, and it can happen at the spinal cord level, that certain types of things will initiate certain types of responses. So in this simple hand withdrawal reflex, information is carried from skin receptors along the sensory neuron to the spinal cord. From there, it's passed via interneurons to motor neurons that lead the muscles in the hand and arm to go away. Because this reflex involves only the spinal cord, the hand jerks away from the candle flame even before information about the event has reached the brain where we experience pain. So we are already acting on that information from the reflex in the spinal cord level before the brain's even aware of what happens. And we'll talk about that a little bit later too, about instances where we can respond to things automatically without our brain knowing about it until it receives the after action report, so to speak. So these types of things can happen, and usually it makes sense because certain things can be very dangerous to our body, and therefore it makes sense that we may have developed a sort of protective mechanism along the way to deal with entering our body carelessly like this, so that we get away from the injury before it becomes too injurious to us. All right, so that sort of ends our little review of the nervous system from that perspective of the neuron and the major components of the nervous system. We'll get back to the brain a little bit later, but let's now talk about the endocrine system and the nervous and endocrine system feedback, because basically the idea is that the endocrine system and the nervous system sort of influence each other, as we'll see. So what is the endocrine system? Well, the endocrine system is a set of glands that produce chemical messengers called hormones. These hormones are very similar to the neurotransmitters that we talked about previously. All right. So the glands that we're talking about, I'll just read them real quickly. We have the hypothalamus. It's also a brain region, but also controls, excuse me, the pituitary gland, which secretes many different hormones, some of which affect other glands. Pituitary gland is sometimes referred to as the master gland. We have the thyroid gland that affects metabolism, among other things. The parathyroids help regulate level of calcium in the blood. Excuse me. The adrenal glands, inner part, helps trigger the fight or flight response that we saw earlier. The pancreas regulates level of sugar in the blood. Testes in males secretes the male sex hormone, and ovaries in the female secretes the female sex hormone. So as these glands secrete their hormones, they travel through the bloodstream. So the endocrine system is seen as the body's slow chemical communication system. Okay, so it's more like snail mail as opposed to the email or chat messaging that we saw with the nervous system. So the slow chemical communication system made up of the set of glands that secrete hormones into the bloodstream. And again, these chemical messengers are manufactured by these glands, travel through the bloodstream and affect other tissues, including the brain. Some hormones are chemically identical to the neurotransmitters that we mentioned earlier. So the adrenal gland, a pair of endocrine glands situated above the kidneys, and they secrete the hormones epinephrine and norepinephrine, and they help to arouse the body in times of stress. Again, that's why they're going to be very important to the fight or flight response that we talked about previously. Then we have the pituitary gland, the endocrine system's most influential gland. It's under the control or influence of the hypothalamus, a brain structure, and the pituitary regulates growth, okay? And it does control other endocrine glands as well. Now, when it comes to regulating growth, I'm sure you guys have seen people who are really short in stature that may be abnormally so, or people who may be 8 feet plus tall, abnormally tall, as far as human beings go. And normally this is probably going to be some issue with the pituitary gland, the gland that regulates and control how well or in what ways that we grow. So some individuals who may have some issue with height, they may get pituitary treatments with the hormones that it releases in order to help stimulate growth in them. Okay, there are certain criteria that need to be met for that, but that is something that does happen. And so we see the pituitary gland is a very significant one that plays a very significant role in how we grow and are develop. And we'll see more about that in a future chapter as well. Now, I want to really do with this slide here sort of a quick comparison between the two systems that we have briefly gone over today, the nervous system and the endocrine system. So first, we have the speed of action. The nervous system, very rapid. The endocrine system can be very slow. All right. The nature of the message, we have electrical impulses traveling along the nerves in the nervous system, and then we have the chemical messages traveling in the bloodstream, the hormones. The duration of the response, usually in the nervous system, it'll be completed within seconds. So whatever it does it somewhat quickly, done within seconds. But for the endocrine system, it may take years before completed. For example, one of the key things that we see the endocrine system involved in, particularly, is going to be puberty. And puberty may be as long as 2 years as your body releases all the specialized chemicals based on your sex for your body to grow, get taller, and develop certain female or male characteristics of an adult. The area of response for the nervous system is often confined to one area of the body. Response is localized, whereas usually in the endocrine system, many organs response is going to be much more widespread. So hormones in the body, widespread impact across the entirety of the body. An example of processes controlled. For example, in the nervous system, as we saw previously, we see reflexes such as blinking, controlled by the nervous system, and movement of limbs. But in the endocrine system, we have the growth when we physically get taller and larger, and the development of the reproductive system. As then, once we go through puberty, as we'll talk more about this later, once we go through puberty, we're then capable of participating in reproduction. All right. Now, as I alluded to earlier, the nervous and endocrine system have a certain feedback. So the brain influences the pituitary gland, and the pituitary influences other glands in the endocrine system that releases the hormones that then will have an impact on our body and, again, our brain. So it's sort of a cycle. So this reveals the intimate connection between the nervous and endocrine systems. Let me walk you through real quickly here a brief example. So let's say we have normal body temperature, homeostasis, we have a balance. Okay, then the normal temperature is disturbed in somehow, and our body temperature rises. Then receptors, okay, temperature sensors in the skin and the hypothalamus detect this rise in body temperature. The information then is going to be sent. The information is going to affect the control center of the brain, okay, the thermoregulatory system, temperature regulating system, and then automatically it's going to send commands to certain types of effectors. Sweat glands in the skin increase secretion, and the blood vessels in the skin dilate until normal temperature is restored. So we see here the nervous system as well as the endocrine system taking part together in this feedback system. So they influence each other. Okay, now the brain. Well, this section that we'll be covering, which is a large section, we'll probably cover only a portion of it. The tools of discovery, the old brain and the new brain, the ideas of brain plasticity, and split brain. So what we'll see in this section, we'll learn about scans and more, about the primitive life-sustaining inner parts of the brain stem and the limbic system, and the higher brain structures that help us think and act, the cerebral cortex. What we normally think of brain, we usually think of the cerebral cortex. Okay, it all starts with understanding what the brain does. And I want to take you back real quickly here to uh, more invasive procedures like lesioning. And basically what this invasive procedure is when you go in there into the brain and destroy a portion of the brain to see what impact it has on the organism. Now, let me describe to you, I might have covered this briefly before, but the idea is how do we discover all this stuff that we know about the brain? But if we can imagine a few hundred years ago, before we had any of this technology, the only way that we really understood the brain could be responsible for some of this is that when somebody behaved somewhat in an odd fashion when they were alive or had some sort of major deficit, and then they died. And then some enterprising young person, physician or something, wanted to find out how does this person's brain differ from somebody else's. And so they would do sort of an exploratory autopsy, crack the skull open, split the brain open, and start to poke around there and see if their brain of this person who had these issues when they were alive differs from another person's brain. Again, we will say this person is dead and they died of natural causes, hopefully, or something like that. And their brain is being observed too. And so they were able to see that after a while, certain areas of the brain of certain individuals who had certain behavioral issues or deficits seemed to be atrophied or not the same as someone who did not have that particular issue. And so they were able to see what areas of the brain might be responsible for certain activities. So that was a very way that we could do things, but again, it would require people to be dead already. Invasive procedures are much more humane in a way that they could lesion parts of the brain, and again, this would most likely be with animals, not human beings, to see how that has an influence on how they process mentally or on their behavior. Now, fortunately for us, the cognitive revolution came along, and we have technology that allowed us to do many, many more things. And we'll see some of these in the next coming up slides. We have the electroencephalogram, the MRI, the fMRI, and the PET, the positron emissions tomography. And I'll describe them a little bit as we go on here. But what they allow us to do is to look inside the brain, so to speak, in some fashion or form to try to figure out what's going on in there. The easiest one is going to be an EEG, the electroencephalogram, where a recording of the electrical waves sweeping across the brain's surface can be detected. So an EEG is useful in studying seizures in sleep. I've had a couple of studies too where I've had the stuff glued onto my scalp. And so normally what happens is they have these electrodes that are placed on the scalp in various locations, allowing us to detect the electrical emissions of your brain as you go through certain levels of consciousness, for example. So they can tell you when you are alert and thinking about something, or when you are falling asleep, for example. And we'll talk more about that when we talk about states of consciousness a little bit later. Then we have the PET, the positron emission tomography. The PET scan allows us to see what part of the brain is active, tracing where a radioactive form of glucose goes while the brain performs a given task. So normally what will happen here, you would probably be hooked up to an IV and they would shoot you up with this radioactive glucose so they can track it as it goes through your brain as you are working on some sort of task. That way they can figure out what part of the brain is involved in certain types of activities. Then we have the MRI, the magnetic resonance imaging. It makes images from signals produced by brain tissue after magnets align in the spin of atoms. The arrows here on this graph show the ventricular enlargement in a schizophrenic patient. So here with this imaging, we can see the ventricles in the middle of the brain. And the difference that we can see here is that one on the right is much larger. And usually when we see larger ventricles, we normally think of the brain as becoming deteriorated in somehow. There could be other reasons why this may happen, okay, but it's not a sign of normality. And then we have the fMRI, or the functional MRI. It reveals brain activity and function rather than structure. So it's not looking for structures about what you're doing. So the functional MRI compares successive MRI images taken at split second apart and shows change in the level of oxygen blood flow in the brain. All right, here are some summary of some of those tools. I just want to cover a couple of them. A few of them may be somewhat newish here. But we have the EEG, where you have the electrodes placed on the scalp. It measures electrical activity in the neurons. And this is what they can find. Symptoms of depression and anxiety correlate with increased activity in the right frontal lobe, a brain area associated with behavioral withdrawal and negative emotion. So this is what some of the research has found with these types of imaging or discovery tools for the brain. Then we have the magnetoencephalograph. A head coil records magnetic fields from the brain's natural electrical currents. Soldiers with post-traumatic stress disorder, compared with those who do not have PTSD, show stronger magnetic fields in the visual cortex when they view trauma-related images. And for PET, again, it tracks where a temporarily radioactive form of glucose goes while the brain of the person given it performs a task. Monkeys with an anxious temperament have brains that use more glucose in regions related to fear, memory, and expectations of reward and punishment. Okay, so we see from a variety of these types of findings how they have been used and may be used for other types of things. The last couple more here, people's MRI, people sit or lie down in a chamber that uses magnetic fields and radio waves to provide a map of the structure of the brain. People with a history of violence tend to have smaller frontal lobes, especially in regions that aid moral judgment and self-control. And finally, the fMRI measures blood flow to brain regions by comparing continuous MRI scans. Years after surviving a near plane crash, passengers who viewed material related to their trauma showed greater activation in that brain's fear, memory, and visual centers than when they watched footage related to the 9/11 terrorist attack. So there seems to be more of a personal connection finding there. Now, we'll see more about these types of things a little bit later as we dive more into the brain and the areas of the brain that seem to have some responsibilities for some of the things like emotions and fear. Okay? Now, there are some new techniques that are being employed as well, tools of discovery, having our head examined here. New activities are focusing on using mapping technologies for a variety of reasons to make new discoveries about how we are influenced by certain things. The Human Connectome Project is using cutting-edge methods to map the brain's interconnected network of neurons. Scientists create a multicolored neural fibers that transport water through different brain regions. So the idea here is that they're getting a really clear, detailed picture of the interconnected neurons themselves. Now, some of the new activities that they are focusing on are such things as neuromarketing, neurolaw, neuropolitics. And the next slide will sort of detail a little bit about what that is. And I did include on your Canvas site a short clip, I think, on one of these. I think it might be neurolaw, I believe, to describe exactly what they're using this area for. Now, this is what this stuff really means. Neuropolitics is a science which investigates the interplay between the brain and politics. So in this example, they could be looking at images of the brain for certain individuals as they talk about certain political things or may hold certain political ideas, for example. Then we have neural law, a field and interdisciplinary study that explores the effects of discoveries and neuroscience on legal rulings and standards. Again, we'll talk briefly about this a little bit later, but neural law is important in that we understand, for example, there are certain defenses out there for if you commit certain crimes and there is something mentally wrong or structurally wrong with the brain, you may not be held fully accountable. And then we have neuromarketing. It's a commercial marketing communication field that applies neuropsychology to marketing research, studying consumers' sensory motor, cognitive, and effective responses to marketing stimuli. So we can imagine that they have uh, consumers slash participants of their studies in certain types of devices that can image the brain as they are being shown certain type of stimuli for marketing purposes to see how it is truly influencing them. Okay. Now, this is where I wanted to stop for today. We have only about 10 minutes or so left, so I will stop at this point. And what we'll be covering on Thursday, when we're back, our next class session, we'll be looking at the brain. We'll be looking at the old brain, the new brain. We'll be looking at a variety of the issues related to the brain, such as plasticity, as well as split brain. That will be our last session before we are going to have a test, okay, which will be covered again, chapters 1, Appendix A, and this particular chapter as well. Now, I'm going to see here, are there any questions or comments at this time? Okay, if you joined us a little bit late, please take the opportunity now to send me a note that you are present. If your name is not associated with your identity that you logged in with, please make sure your name is present. Include it when you send me the statement that you are present here today. Okay, great. So what we're going to do, I'll be putting together a study guide for you guys a little bit later that I'll post as you can use to help prepare you a little bit more for the upcoming exam. If there is nothing else, I'll let you guys go now, and I'll see you on Thursday. All right, bye-bye.